Hi everyone, I'm Dora and this is the biology video for cell structure. Now we can begin with saying that cells are the basic structural unit for all living organisms. We know that some small organisms only consist of a single cell and they're called unicellular organisms, and larger organisms which consist of more than one cell are called multicellular. So the statement that living organisms consist of cells is an example of a theory, and this theory is the cell theory, which was developed by Robert Hooke. Now we can get on with microscopy skills. So we know that microscopes use lenses that allow us to look at structures that are too small to see with the naked eye. A single convex lens is useful for magnifying up to 20 times, but this is not enough for studying the structure of cells. So we use microscopes with 2 or more lenses to be able to see much smaller structures as the magnification of the lenses are multiplied. This is the magnification formula, which is the size of image divided by the actual size of specimen. Now we can get on with developments in microscopy. So microscopes were first invented in the 17th century. And obviously, as you can see here, there have been many technological developments in microscopy throughout the years. At magnifications of more than 400, it becomes increasingly difficult to produce a focused image with a light microscope, which is this one right here. And this problem was overcome by the development of microscopes that use beams of electrons instead of light. The wavelength of electrons is much shorter than the wavelength of visible light. So electron microscopes can give magnifications up to 1,000,000. And then we have some examples of electron microscopy. The first being fluorescent stains and immunofluorescence. So basically most of the chemicals in cells are white or colorless, so for us to be able to see them, they need to be stained. And this technique uses stains that bind to some chemicals but not others. So for example, methylene blue binds to DNA and RNA, so it stains the nucleus dark blue and cytoplasm a lighter blue. Fluorescence is when a substance absorbs light and then re-emits it at a longer wavelength. So by using this, we can generate particularly bright images. A multicolored fluorescent image can then be produced showing where the chemicals are located. You can see in this one. And then we have freeze-fracture electron microscopy. So this technique is used to produce images of surfaces within cells. So a sample rapidly freezes. As you can see here, this is the first step. And this creates a replica of the fracture surface. It's like an outline that you can see here. It's replicated. This is an example of freeze-fracture image of a yeast cell. And as you can see here, many different components of the cell can be seen, such as the vacuole, the cytoplasm, a lipid droplet, and the plasma membrane. So the next technique is cryogenic electron microscopy. And this one, it's basically used for researching the structure of proteins. And so the steps are basically they freeze a protein solution, and then the solution is placed in an electron microscope, and the pattern of electrons are detected. And then using computational algorithms, these patterns are combined to produce a 3D image of the protein molecules. These are some of the examples of cryogenic electron microscopy. We have the plasma membrane, cytoplasm, and DNA. So the plasma membrane is the outer boundary of the cell and encloses all of its contents. The cytoplasm consists of water and There's still many substances dissolved or even suspended in this water. Enzymes in the cytoplasm catalyze hundreds or even thousands of different chemical reactions, and these reactions are called the metabolism of the cell. Then we have the DNA, which is the genetic information. Genes made of DNA contain the information needed for a cell to carry out all its functions. So many genes hold the instructions for making a protein, and some proteins are structural, so are needed for growth and repair. Others act as enzymes and catalysts. Then we have the structure of prokaryotes. Prokaryotes were the first organisms to evolve on Earth, and they still have the simplest cell structure. They're mostly small and found everywhere. In soil, water, in our skin, our intestines, and even if in pools of hot water in volcanic areas. So we said that all cells have a plasma membrane, and some cells, including prokaryotic cells, also have a cell wall. Prokaryotic cells don't have a nucleus, so the interior is entirely filled with cytoplasm. The cytoplasm is not divided into compartments by membranes, instead it's one uninterrupted chamber. So they're much simpler than eukaryotic cells. Prokaryotes do not have cytoplasmic organelles apart from ribosomes. And prokaryote ribosomes are smaller than those of eukaryotes. There are 70S in prokaryotes, and we have 80S in eukaryotes. The eukaryote cell structure. We have the nucleus, which is the compartment holding the cell's chromosomes. The nucleus has a double membrane, which is important, with pores through it. Each chromosome consists of one long DNA molecule attached to the proteins, which you can see here, the histone proteins. We have 80S ribosomes. The ribosomes synthesize proteins, but there are structural differences and they're larger in size, as we said. We have the mitochondria, the powerhouse of the cell. So a mitochondrion is surrounded by a double membrane. The inner membrane is usually folded inwards to increase the surface area. And mitochondria carry out aerobic cell respiration. So in eukaryotes, they're only lacking in cells that never respirate aerobically. The processes of life in unicellular organisms. Homeostasis refers to the maintenance of a constant internal environment in an organism. Metabolism is the sum of all the biochemical reactions that occur in a living organism. Nutrition is supplying the nutrients required for energy, growth, and repair in an organism. Excretion is the removal of waste products of metabolism from an organism. Growth is an increase in size or number of cells. Response to stimuli is perception of stimuli and carrying out appropriate actions in response. And then we have, lastly, reproduction, production of offspring either sexually or asexually. So here we can obviously see different parts that are specialized to perform different tasks. In response to changes in the environment, so this is response to stimuli, whip-like cilia. You can see here these guys are cilia. We have the food vacuoles that contain smaller organisms that the paramecium has consumed. These are digested. These are nutrition needed for growth and energy. So here in the cytoplasm, it shows we have the metabolic reactions occurring. Also, enzymes in the cytoplasm catalyze these reactions. This big thing is the nucleus of the cell, and it can divide to produce the extra nuclei needed when the cell reproduces. This is the cell membrane, and it controls what chemicals enter and leave. This here is a contractile vacuole, and they fill up with water and then expel it through the plasma membrane. which is a type of homeostasis, keeping the water content of the cell within tolerable limits. And this is a plant cell. It has a cell wall, which is freely permeable, contractile vacuoles, two flagella, nucleus, food vacuoles, and we have a chloroplast, and also photosynthesis occurs in the chloroplast. From this table, you can see the differences in eukaryotic cell structure between animals, fungi, and plants. Atypical cell structure in eukaryotes. So, for example, we can give red blood cells who do not have a nucleus, phloem sieve tube elements, skeletal muscle, and aseptate fungal hyphae. You can study on this table for the structure and functions of all the main organelles of eukaryotic cells, which are nucleus, rough ER, smooth ER, Golgi apparatus, lysosome, mitochondrion, free ribosomes, chloroplast, vacuoles and vesicles, and microtubules and centrioles. Also, cytoskeleton and cilia and flagella. This is the electromicrograph of a liver cell, and as you can see here, the plasma membrane is visible as a dark line. This is the mitochondrion, this is the Golgi apparatus, this is the lysosome, and this is rough ER. So let's go on to the origin of eukaryotic cells by endosymbiosis. In endosymbiosis, one organism lives inside another, which is called the host. The endosymbiont enters the host cell by endocytosis. This is the process that cells use to make a vesicle or small vacuole by pinching off a piece of the plasma membrane. Cells can use endocytosis to ingest other and smaller cells. For example, phagocytes in humans ingest viruses or bacteria, and unicellular organisms such as paramecium ingest the organisms on which they feed. So this is theoretically the origins of the nucleus, mitochondria, and chloroplast. Because they have a double membrane, it can be theorized that they were formed through endosymbiosis. These are the features of both mitochondria and chloroplasts that provide strong evidence for the endosymbiotic theory, which is summarized here. They have a double membrane. This would be expected if a prokaryote with a single plasma membrane was ingested by endocytosis. They have their own genes on a circular DNA molecule like that of prokaryotes. They transcribe their own DNA and use the mRNA to synthesize some of their own proteins. And the ribosomes they use for protein synthesis have a size 70S and a structure more typical of prokaryotic cells rather than eukaryotic. And they can only be produced by division of pre-existing mitochondria and chloroplasts. Now we have cell differentiation, which is the process for developing specialized tissues in multicellular organisms. So multicellular organisms have an advantage because the cells can develop differently to perform different functions. Specialized cells develop only the features they need to carry out their functions, which makes them more efficient. So for example, we can say red blood cells transport oxygen using the protein hemoglobin, and they produce large amounts of this protein but do not produce other proteins that they would not use, which makes them more efficient. And this development of specialized cell types begins from a very early stage in the life of humans. Even in a tiny embryo, different cells begin to take different pathways of development and differentiate. So in differentiated cells, different genes are switched on and expressed, so the cell makes particular proteins and other gene products. And this control is called gene expression. The evolution of multicellularity. So there are several advantages to multicellularity. They have longer lifespans, they're generally larger than unicellular organisms, so they can exploit niches that single-celled organisms cannot. And they're more complex as there can be differentiation of cell types within an organism, as I've said before. This is the end of the topic cell structure. Thank you for watching this video. Hope it was helpful.